Okay, students. So, in continuation to our lecture on membrane structure and properties, right? In continuation to that, today I'll talk to you about membrane fluidity. Now, once we had discussed the structure of the plasma membrane and its basic functions, I told you that the membrane, although it performs the major function being the outermost boundary of the cell, but this boundary is not so rigid, right? It is fluid in nature. We have also talked of this term in the fluid mosaic model also, right? So now we have already discussed that what is actually fluidity. So now today we'll talk about what are the basic components that actually make the membrane fluid and help the membrane perform its major function of transport of molecules and ions across the membrane. So, when we talk of membrane fluidity, we must keep one thing in mind that membranes are not static sheets of molecules that are locked rigidly in place. I just mean to say that whatever components the membrane is formed of, maybe it is different type of proteins or lipids or carbohydrates. It is not like that. These sheets are or these components are very static and very rigid. It is the movement and flexibility in these molecules that actually decides membrane fluidity and thus determines what are the functions, I mean, especially the transport functions that a particular membrane can perform. So, a membrane is held together primarily by hydrophobic interactions, which are much more weaker than covalent bonds. Now, you know that these covalent bonds are very strong, very difficult to break, and not so flexible, right? Now, for membranes to be flexible and fluid, that is, for membranes to allow transport of molecules across them, or in order to connect these membranes from their external environment, these components are primarily held together by weak hydrophobic interactions. Then, phospholipids, which are present in the membrane, these can also move within the plane of the membrane. You had seen in the diagram where I had shown you the lipid bilayer that a membrane is composed of a number of phospholipids that lie adjacent to each other in one leaflet and opposite to one another in the other leaflet. And once we had talked of the properties of membrane phospholipids, I had told you that they can carry lateral as well as flip flop movements. So these movements also add on to the fluidity of the membrane. Then, of course, temperature affects the fluidity of the membrane since the membrane is mainly formed of lipids. It is a bilayer of lipids, and you know, lipids being the molecules that that have immense effect of temperature on them. You have seen how fats at high temperature they melt, and whereas at low temperature they solidify. Oh, okay. So lipids are drastically affected by temperature. So similar is the case with the membrane, and of course, its overall composition is also affected with this temperature. So we'll just see once I'll move ahead with this topic. We'll see what are the different components that actually add on to this. Then a membrane remains fluid as temperature decreases until finally at some critical temperature the membrane solidifies. Now you see, whatever living system it is, it has its own body temperature, has its own moderate temperature, right? So these membranes normally they remain fluid. I mean, they are not freezed, they are functional, they allow transport across them at these temperatures. And slowly, as the temperature goes below normal, right? These membranes, they it is not like that they become very rigid. They remain fluid, but after some time, I mean, when the temperature is very low, again, it is different for each and every membrane. The membranes can solidify also. In that case, that cell will no longer remain viable or will no longer be able to perform its functions because its membrane is going to be rigid. So, did you get this? Do you have any questions on this? Do you agree to all the points that I have discussed here? Okay, fine. Let's move ahead. Now, how fluid? I mean, when we say a membrane is fluid, it is flexible. Then there must be a criteria to decide. There must be some factor that how fluid or how unfluid a membrane can be. So, how fluid a lipid bilayer is at a given temperature. It depends on its phospholipid composition, in particular on the nature of hydrocarbon tails. We have already discussed this in terms of saturation, in terms of the degree of saturation or unsaturation in the hydrocarbon tails. The more saturated the lipids are going to be, right, in the hydrocarbon tail, in the phospholipids, the more you'll see the rigid the membrane is going to be because of the gaps or the kinks that are lost due to the presence of the double and triple bonds due to unsaturation. We have already discussed this, so this applies here also. Then the closer and more regular packing of the tails, the more viscous and less fluid the bilayer will be. Now this is true, we have already discussed that. Close packing of tails makes the membrane more viscous and hence less fluid. Two major properties of the hydrocarbon tails affect how tightly they pack in the bilayer. I mean, they're going to decide how tight or loose the packing between the hydrocarbon tails and thus the phospholipids of the membrane will be, and thus they will decide that whether the membrane is going to be fluid or not. So the first factor that comes into play here is the length of tails. It is seen that short tails have tendency to increase fluidity, and the second factor that comes into play is the degree of unsaturation. Right? If we talk of saturated phospholipids, then of course the length of tails will come into play. And if we are talking of unsaturation, lipid bilayers that contain a large proportion of unsaturated hydrocarbon tails are often seen to be more fluid than those with lower proportions. Right? So, did you get this? We have discussed it in membrane phospholipids also. So just keep in mind these two factors, the length of tails and the degree of unsaturation of tails. Then comes another factor, that is cholesterol. Now you know cholesterol is present in animal cells, and here in this diagram you can see that cholesterol is found in the cell membrane of animals and it lies between these phospholipid molecules. Now we'll see how cholesterol affects the fluidity of the membrane. So the steroid cholesterol is wedged between phospholipid molecules in the plasma membrane of animals, which helps to stabilize these membranes. It has been seen that at relatively warm temperatures, for example at 37 degrees Celsius, which is the body temperature of humans, cholesterol makes the membrane less fluid by restraining the movement of phospholipids. I mean, each and every membrane has a degree of fluidity. So at these warm temperature, it maintains that fluidity and it assures that the membrane does not, there are no gaps in the membrane and there is no excess fluidity with the membrane. And also because cholesterol hinders the close packing of phospholipids, it also lowers the temperature which is required for membrane to solidify. Right? So at lower temperature also, it is providing protection so that the membrane should not solidify and lose its fluidity. Now, why fluidity is important? First of all, it allows membrane proteins rapidly in the plane of bilayer, right? It holds those proteins, right? These proteins also, they perform a lot of functions. So this is also important for this particular factor. Then it permits membrane lipids and proteins to diffuse from sites where they are inserted into the bilayer after their synthesis. We have already talked of this, that all these phospholipids are synthesized in the cytosol, and they are first incorporated into the inner leaflet of the plasma membrane, and then through flip-flop movements, right, they are transported from the inner to the outer leaflet. So if the membrane is not fluid, not flexible, that is, if it does not allow the movement of phospholipids within its layers, then then to maintain the correct composition of phospholipids in the bilayer would be very, very difficult. Then it also enables membranes to fuse with one another and mix their molecules. Once we will talk about the, once we'll talk about transport across membrane in terms of vesicle formation. I think I had introduced you with this once we had talked of the structure and organization of the Golgi apparatus and the endoplasmic reticulum. So there it has been seen that, and once you learn the mechanisms, you'll see to deliver a cargo, sometimes these membranes have to fuse and form a channel so that that particular protein is transported from a particular, you can say, vesicle to that part of, you can say, any organelle or any of its destination. So you'll see that these plasma membranes, through endocytosis, they receive a lot of material, and it occurs only because lipids of the membrane and the vesicle that is carrying it, they are able to fuse together. Then it also ensures that membrane molecules are distributed evenly between daughter cells when a cell divides, right? We have seen that how important these lipids play an important role, especially in cytokinesis in case of animal cells where furrowing occurs, where these membranes fuse at the equatorial plate and allow the two daughter cells to separate, right? So, are you comfortable with all these? I mean, did you get all this? And do you agree to all these functions? We have already discussed about all these functions in earlier lectures. But the point is, once we learn about membrane, it is very important for us to sum up all this. Okay, let's move forward and talk of the last term that is associated with the structural properties of the membrane, that is called membrane asymmetry. I hope you understand the term asymmetry. Say, for example, if I say in English, what do you mean by the term asymmetry? Okay, Shraddha says irregular. Please, I want a response from everyone. What do you mean by asymmetry? We say that the plasma membrane is asymmetrical. Okay, not equal on both sides. Okay, I'll take you back to the structure of the plasma membrane, right? Which we have talked of. Right. Okay. So now I'll just zoom it a bit. Now you see, this is one side of the lipid bilayer, that is called the extracellular phase, and this is another side of the lipid bilayer, that is called the intracellular phase, because it is facing the cytosol and this one is facing the extracellular fluid. Do you think that both sides of the membrane appear symmetrical to you, or do they look same in terms of their biochemical composition? Please, I want a response from everyone. No, of course. This is because, first of all, the composition of phospholipids in the inner and the outer leaflet is not same, right? You'll see more of negatively charged phospholipids are seen in the inner leaflet as compared to the outer leaflet, first of all. Then, if you take a look at its structure, you will see the composition of cytoskeleton also varies on both the leaflets, right? The composition of protein also differs in case of both the leaflets, and the carbohydrates are seen only on the outer leaflet of the membrane, and you don't see any carbohydrates in the inner leaflet. That is, you'll see most of the glycoproteins and glycolipids on the outer surface of the lipid bilayer, and you'll see the extracellular matrix that, of course, is covers or is seen outside in contact with the outer leaflet of the membrane. So all these things clearly indicate that the membrane is asymmetrical, and this asymmetry is very important to perform all the functions that are required by the plasma membrane. And of course, these lipid bilayers, they appear to be one, but you'll see once you learn more of cellular structures that actually the inner and the outer leaflet, they are capable of performing different functions also because of these different compositions. So definitely the plasma membrane is said to be asymmetrical. So we'll just go back to the term membrane asymmetry. So the plasma membrane is said to be asymmetrical because the membranes have distinct inside and outside faces. First of all, the two lipid layers differ in specific lipids compositions. This I have already told you. The two lipid layers differ in protein composition, and also the plasma membrane has carbohydrates which are restricted to be present only on the exterior surface. So it might have proteins and phospholipids in common in both the leaflets, that is the outer and the inner leaflet, but their type and composition varies. Okay, so this is all about the structure of the membrane that we have to talk of. We'll continue with membrane functions. Okay, so I'll just take a pause, and in the next talk we'll talk about the specific functions of the plasma membrane.